we're going to end our lab technique video with a discussion of proteins. And proteins, luckily, are easier to work with than RNA. They're a little bit more stable. So in the same way that we can use an electrical current to separate DNA by size, we can do the same thing with proteins. Proteins have some challenges, though. First, unlike DNA, they're not all negatively charged. And second, they have a three-dimensional shape to them that's going to affect how they move through a gel. A 300 kilodalton fibrous protein that's long and skinny is going to move faster than a protein that is more round and globular that's maybe the same weight, because the skinny one can squeeze through the matrix better. So to overcome these challenges, we're going to first treat our samples with a few things. First, we're going to treat them with SDS, and this is going to coat your protein in a negative charge. We're also going to add a reducing agent, and this is going to break up any strong disulfide bridges in between those cysteine residues. We're going to get rid of those. And then we're going to boil it. And the heat is what's going to denature your protein and make it lose all of its fun three-dimensional shape, its tertiary and secondary structure, all gone. So once you've treated your sample, you're ready to load your gel. You're also going to load, same thing we did with the gel electrophoresis for DNA, we're going to load a molecular weight ladder so we know what the sizes are. We're then going to add our samples that have been treated with this SDS, this reducing agent, this heat, also a loading dye so we can see it, and we're going to add them to the wells on the top of our gel. We're then going to bathe the gel in buffer, and we're going to apply an electrical current. And again, those small proteins are the ones that are going to be squeezing through. So they're going to be attracted to the positive charge at the bottom, and they'll move faster through the gel. Now we can take that gel and we can apply a stain like we did for DNA, and that will show us all of the sizes of proteins in our sample. It's not always the information we want, though. Proteins have a special relationship with antibodies that allows for a more specific detection. And this we call a Western blotting, or you sometimes refer to as immunoblotting. Antibodies recognize a specific antigen, which is a specific peptide sequence. And they can be made to bind to your protein of interest. So say you're interested in actin, again, that cytoskeleton protein. Well, there are antibodies that are commercially available that recognize just actin. They're specific for actin. So after we run our SDS page gel, we can then take the proteins and transfer them to a nylon membrane. Because the gel, what happens over time is your proteins are eventually going to move out of the gel once you stop applying an electrical current. So we're going to do a transfer setup, and that's what's shown here. This is kind of an old-fashioned way of doing it with liquid. These days you can do a dry version that's much faster, but the same principle applies. You're going to use electricity, and again, that the proteins are still with that SDS, they're still going to be negatively charged, so they're going to move towards the positive electrode, and you can apply an electrical current to move your proteins from the gel into a nylon-based membrane. This membrane is much easier to work with. It's like a little piece of kind of slimy paper, I guess I would say, thin slimy paper. So then you can expose your nylon membrane to a primary antibody, which is going to bind to your protein of interest. Again, we have that actin antibody. It's going to bind wherever actin is on that membrane. And you can follow it by a fluorescent antibody, an antibody that's a secondary antibody. So its antigen, its recognition sequence, is going to be for antibodies in general. So the secondary antibody will bind to wherever that primary antibody was laid down. And then we can image it. Now this is showing the old-fashioned way of doing it with film, so like camera film, and I've done this before. You go into a dark room and you play with all these like chemicals to develop film like an old-timey photographer. These days we don't mess around with that anymore. Instead, we're just going to use fluorescence, and you're going to take a picture with a camera. It's a fancy camera, it's a fluorescent camera, but you just image your gel or your membrane, and it will have bands that will show you where your protein is. You can check the size, and the thickness of the band will tell you about how much protein was in your sample compared to other samples. Okay, so if we wanted to isolate a particular protein from a sample, We can also use chromatography. So in this technique, we're going to run a sample through a column that has beads on it. And those beads either bind charged things, or they trap small things if it's a size-based column, or sometimes they have antibodies on them, so they'll bind a specific protein. Depending on what you're trying to do, that will determine what kind of beads you're going to put in your column. But when you wash your sample through, your protein is going to bind to the beads depending on what kind of beads they are and what you're studying. And you'll collect all of these fractions. And that way you can isolate your protein from a complex mixture. Finally, say your goal was to look at organelles. So you wanted to know specifically about the mitochondria. So to do this, we're going to take advantage of centrifugation, which is where you spin things, and heavy things are going to fall to the bottom and lighter things are going to speeds. You're going to isolate smaller and smaller organelles. So you see, you start with your sample, you're going to break it up. We call that homogenize. You're going to homogenize your sample, make it kind of like mush. And then you're going to put the mush in a tube and you're going to spin it first at a slower speed. The biggest things like nuclei, they're going to come to the bottom, and everything else is going to remain in like a liquid phase. You increase the speed from 1,000 Gs to 20,000 Gs. Now we're going to get medium-sized things. You keep doing this, you're going to get smaller and smaller molecules. And in this way, you can separate out your organelles. So in this video, we discussed the last of our basic cell biotechniques. Again, we're going to come back to most of these in the lab modules.